So I hope all the evolutionists that are watching this, I hope you can realize that you just found a case that you should break down the evolutionary lie and embrace the truth.
The same day, all the fountains of the great deep were broken up, and the windows of heaven were opened. Welcome to this episode of Hydroplate Theory University with Hydroplate Theory experts Kevin Lea and Bill Wells.
Well, hello everyone. My name is Bill Wells, and along with me, Kevin Lea. Welcome to our Hydroplate Theory University 9th podcast. This will be presented on August 19th, 2026. And our goal is to teach through the 9th edition of Dr. Walt Brown's Hydroplate Theory. That book can be purchased online as well as read for free on the CSC website. The links will be below. In this episode, we are continuing the first episode where we are answering questions submitted by the viewers about the asteroid introduction parts 1 and 2, the episodes dealing with the overview of the asteroid chapters. And the answers are taken from Dr. Walt Brown's book, In the Beginning, Compelling Evidence for Creation and the Flood. We like to start off with kind of our key verse describing what the Bible says actually happened during the flood from Genesis 7:11 through 12. In the 600th year of Noah's life, in the 2nd month, in the 17th day of the month, on that day, all the fountains of the great deep were broken up, and the windows of heaven were opened. And the rain was on the earth 40 days and 40 nights. In the Genesis 1 account of creation, it says that God separated the waters above from the waters below with the firmament. Dr. Brown makes a good case that the firmament was the upper crust of the earth. So the pre-flood situation would have a sandwich effect, basically. The pre-flood seas on top of the surface with about 60 miles of spherical shell of granite. And then below that, spherical shell of water, completely sealed in by the granite shell, and it would be a little over a mile thick. Literally another ocean of water. And as physics takes over, the water heated up, became supercritical, began eating away at both the ceiling and the upper granite crust and the lower mantle floor, in which the pressure increased and a crack opened up, and that was the fountains of the great deep bursting forth, which followed created a chain of events which is understandable by known chemical, physical laws and processes in cause and effect relationships. So in this episode, we're going over a third question or statement from looks like Dr. Feldman again, from our YouTube channel. These questions are taken from the YouTube channel, and he states, there's no traces of plants or bacteria, only chemicals, which may or may not have come from life. But as taught in part 2, intro to the asteroid chapter, which is podcast number 6, they did find 14 of the 20 amino acids necessary for making proteins on Bennu and Ryugu, and also bacteria on the asteroid Ryugu, which we'll be talking about today. So 14 of the 20 amino acids necessary for making proteins. Evolutionists and non-hydroplate theory creationists, unfortunately, we have many within the creationist camp that contend that these 14 amino acids are the result of chemicals randomly forming amino acids in space, and not from life. They claim it comes abiotically. They make this argument based on the fact that amino acids that come from live proteins are left-handed, but the samples from the asteroids were equally left and right-handed. Now we're going to be getting into a discussion of amino acids. There are probably many of you that already know about amino acids and proteins. Some of you may not. So to get the audience up to speed, we'll be talking about amino acids, what they are, why they're necessary for making proteins, how they're made, and eventually we'll get down to this issue of both left and right-handed proteins, or left and right-handed amino acids. But first, we're going to cover some basics to give you some framework to be able to better understand the significance of finding amino acids and how to sort through a correct interpretation versus a false interpretation. So some facts concerning amino acids. Why does it matter? Cellular machinery like ribosomes and enzymes are being directed by DNA instructions. They act like specialized locks that only accept the correct molecular key. Using wrong-handed version would disrupt vital processes like protein folding and DNA replication. So to try to help us understand the big picture about amino acids and proteins, when I started teaching this a number of years ago, I decided that Legos would be a good teaching tool because Legos are familiar to help us understand something that's not familiar. And so we all know we need protein to eat. And so, for example, when we eat cow protein, a burger or steak, your body really actually cannot use the proteins that you're eating. And in fact, if some of those proteins were to be released into your bloodstream, your body would set up an immune reaction. That's called leaky gut, where the intestines allow proteins, undigested proteins, in the bloodstream and create immune reactions like eczema or asthma. So you cannot actually eat the cow proteins. So cow proteins, we're going to call those cars, Lego cars. And the parts that you have available in those Lego cars, those are going to get smashed down into their component Lego parts by the stomach acids and enzymes. Now, in all living things use the same selection of 20 amino acids. And in this case, in the example, they're all red and they're all different parts. And they're linked together and folded to make an enormous amount of different proteins for all living things. So the digestive system breaks the Lego cars down into the component little pieces of Legos. And if everything is working right, there's little perforations within the intestinal wall that allow these little tiny Lego parts, the amino acids, to go into the bloodstream. The bloodstream then carries those amino acids to all the cells, and the cells, they do grocery shopping, so to speak. They bring in all the groceries, they bring in all the, not to mix metaphors, but they bring in all the Lego parts, a complete selection of many volumes of these Lego parts. So using the amino acids and proteins Lego analogy, our bodies have well over 100,000 different proteins. And in this analogy, each protein that your body uses is a Lego airplane. And each airplane, each of those 100,000 different protein airplanes are built from a selection of just 20 different amino acids, those that were provided to the cells by the digestion of the Lego cars, cow protein. And then your cells will arrange those in different ways to produce the over 100,000 Lego airplanes that your body needs, the proteins. So using the directions found in the DNA, the cells build the needed Lego airplanes, which you can use, and they do that from the parts that used to form Lego cars. And so amino acids, you can see, are the building blocks of life. And it is through the direction of the DNA and cellular machinery that takes that information, the cell sort of goes shopping through all of its supply of the 20 different amino acids and starts grabbing them and linking them together in long chains, sending them to something called a chaperone, which then folds that chain into a specific shape. And then those new proteins, those new airplanes, are now shipped to the other parts of the cell that need a replacement or a new or duplicate protein.
And there's there's many animations that have come out over the years from microbiologists that show this process. And so this is taken from YouTube. It has copyright information, which I'm complying with the copyright information here by letting you know that this is the YouTube, and it's animated by Polyamine Animation Company, but it's then actually they did it for yourgenome.org. And by letting everybody know that's where it came from, we are then able to show this incredible animation of what happens inside of your cell.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
So, after watching that, how could anyone, including any evolutionist, any professor of microbiology, genetics, all of them, how could they believe that extremely complex, engineered, biological system contained in every living cell could have happened by time and chance? I mean, you have the DNA that sends the instruction, and what we're going to emphasize, the DNA tells the RNA, you have to go get red Lego amino acids, which are left-handed. You can't go get blue, as we're going to see, Legos that are right-handed, because all proteins of living things are made out of left-handed amino acids. And so it tells the RNA, just go get left-handed, and it cuts the RNA to say, okay, well, we're going to be making, the DNA tells the RNA, we're going to make this kind of a protein. So they have to cut out the parts that aren't necessary, band it together, and then those messengers are supposed to go out there and look for the proper amino acids that have been delivered to the cell through the blood system, circulatory system, and then put it into a protein that gets folded up in just a perfect way. If it doesn't fold up right, it's cancerous, causing it causes problems. It's not able to be used. And they think that happened by time and chance. Only the evolutionists think it's time. I think an extreme intelligence, all-knowing, all-powerful, extreme intelligence created life. And that life is allowing us as human beings with a soul to decide who we want to serve, the creator or the creation. Answer, the only ones that can be thinking this is time and chance are religiously devoted to their time and chance God. And this is what Darwin said. If it could be demonstrated that any complex organ existed, including every single cell in your 60 trillion cells in your body, which could not possibly be formed by numerous successive slight modifications, my theory would absolutely break down. But I can find no such case. I wish we could go to his grave, clone him, put the soul back into him that's left and waiting for judgment, and say, well, what about now? At the time in the 1800s, you didn't know about biology in the cell of a human being. Now, with what we just showed, what do you think? And I think, you know what he would say, especially since he'd be coming from the dead. He'd say, I want to burn every book that ever got published as a result of my foolish analysis of the origins of the species. Facts and terms pertaining to amino acids. Why don't you go for this? This is your expertise, Bill.
Thanks, Kevin. So now we've had a pretty good understanding that proteins are really, I guess you could call them the building blocks of life. But those building blocks have building blocks, and they're called amino acids. And so having laid that basic understanding, we're now going to introduce you to something that just lets you know that amino acids, not all amino acids are the same. And Kevin kind of referenced it. There are left-handed amino acids and there are right-handed amino acids. And living things only use the left-handed amino acids. So DNA uses the left-handed amino acids to build the protein. It's called chirality. And so chira comes from the Greek word hand. And so you have left-handed and right-handed amino acids. Many molecules, including amino acids, exist in two mirror image forms called enantiomers, much like your left and right hands. Your left and right hands are mirror images of each other. And this process is known as chirality. Now, left-handed amino acids, living organisms on Earth almost exclusively use the L configuration of amino acids to manufacture proteins. Out of the 20 common protein-building amino acids, 19 are left-handed. One of them is not chiral, meaning it does not have handedness, and that's glycine. So homochirality is a strict preference that living systems have for one single-handedness, in this case the left-handed amino acids. In biology, this is called homochirality. For comparison, the nucleic acids in DNA and RNA are built using only right-handed sugars. So Frank Hubeny had another great answer to Dr. Feldman's question about no living things being found on the asteroid. And he says this: Jonathan Sarfati, who's with Creation Ministries International, wrote, "Were life's building blocks found on Bennu, this December 2025 edition, which provided a detailed summary of Bennu, the OSIRIS-REx mission, and what was found. Dr. Sarfati did not reference the hydroplate theory, although he should have. The amino acids that were found on Bennu were racemic, and that means they had a 50-50 mix of left and right-handed amino acids, which bothered me at first until he mentioned that homochiral amino acids, the left-handed only ones, would become racemic 50-50% left-handed, right-handed after thousands of years. The two papers on the results, one in Nature and one in Nature Astronomy, are open access and referenced in Dr. Sarfati's article. So here's an illustration of left and right-handed amino acids, and this is a NASA photograph taken about a description of the Magnuson meteorite, in which many of amino acids were found, both left and right-handed.
If you're enjoying Hydroplate Theory University, be sure to subscribe to our YouTube channel by searching Hydroplate Theory University and clicking our official logo. We're organizing the series into playlists for each chapter of Dr. Walt Brown's book. Right now, we're covering the chapter overviews, and once those are complete, we'll come back and explore each chapter in greater depth. You can also find every episode and chapter at hptuniversity.com. We're still finishing a few parts of the website, but everything will be there soon. Now, let's get back to Hydroplate Theory University.
Up until this point, we have only been talking about amino acids from things that were living and then instantly disassembled when the fountains of the Great Deep burst open and launched them, along with all the other asteroid-forming materials, into space. So most likely all those amino acids from living things would have been left-handed when they became parts of asteroids Ryugu and Bennu, and then began the process of transforming into collections of left and right-handed ones called racemization. But there would have been another source that would have provided ready-made mixtures of both left and right-handed amino acids. As per my example, on Earth before the flood, there were already pre-made piles of both red and blue Legos, ready to be launched into space and gently settle onto asteroids like Ryugu and Bennu. That supply of already racemized amino acids would be provided by the millions of living things that had died before the flood began. Just before the 46,000-mile-long crack opened and subterranean waters burst up, there also would have been a massive supply of both left and right-handed amino acids from millions of dead people, plants, and animals that were in various stages of decay. Now, people, plants, and animals have been living, growing, dying, and decomposing for over 2,000 years by the time the flood began. When living things die, the decomposition process begins, and it's a process which involves their component amino acids beginning to racemize, change into left and right-handed amino acids. The right side of this picture depicts amino acids that are racemized, now left and right-handed ones. To illustrate this using our Lego example, the once collection of pure left-handed amino acids on the left, the all-red Lego group, begins to change into a pile of both left and right-handed amino acids when the living thing dies. Over time, the amino acids continue to decompose down to their component molecules to be recycled, largely by plants, into new left-handed amino acids. But for a long period of time before complete decomposition, there would have been a massive number of piles, so to speak, of racemized amino acids. The dead things would have been subjected to the same forces as the living things. But in varying stages, the decomposition process would have already done most of the disassembling of the proteins, leaving collections of left-handed amino acids to begin the decomposition process. This well-known decomposition process, in which proteins are broken down to their component all left-handed amino acids, which in turn racemize into collections of both left and right-handed amino acids, is the basis for a dating method called amino acid racemization dating. Search AI defines it this way. Amino acid racemization dating is a technique used to estimate the age of biological specimens by measuring the conversion of amino acids from their left-handed forms to right-handed forms after the organism's death. This method is useful in fields like paleontology and archaeology to determine the time lapsed since the specimen was formed. Now, I'm referencing amino acid racemization dating only as a way to demonstrate that this racemization of amino acids in dead things is a very real-world phenomenon, and not suggesting this is a way to determine the ages of asteroids. That dating method would be impossible on asteroids because all of the materials caught up in the fountains of the great deep would be quickly and randomly mixed together as they exited the Earth's atmosphere in just a few seconds on their way to exiting Earth's gravity well and into orbit around the sun. So, to summarize, the hydroplate theory can easily explain that the left-handed and right-handed amino acids found on asteroids Ryugu and Bennu came from Earth, and that they are the racemized amino acids from both living and dead things launched during the Genesis flood. Now, following the science behind all of this, I would like you to consider a very strong implication arising from what science observes and knows about amino acids. I asked AI a couple of questions that gave some interesting answers. Question 1, are right-handed amino acids required for any life functions? Answer, currently, there are no observable life functions that require right-handed amino acids. The second question took me a while to finally get there to define the abiotic source coming or producing right-handed amino acids as only out in nature. But I asked AI if the only known abiotic source of right-handed amino acids was their conversion from left-handed ones. And after clarifying that I meant an abiotic process observed in nature, I got this answer. You're right. I conflated direct natural observation with experimental demonstration of chemical possibilities. Under your stricter definition, the direct formation of D amino acids, that's the right ones, by abiotic process has not been observed occurring in nature. So, to summarize that, as far as what science now knows, right-handed amino acids are not necessary for life, and they are only formed out in nature from existing left-handed ones. So putting these together as a creationist, I have to ask this question. Why did God make amino acids, left-handed amino acids, so that when found outside of living things, they racemize into a 50-50 mixture of left and right-handed ones? In my opinion, God did that to provide yet another message written to those living the age of biochemistry. This message screams at us that life did not evolve from the actions of the God of time and chance, but rather life was created by God, as he told us in the Bible. To create life, God did not need to make right-handed amino acids, but he did. God most likely did this at the time of the fall of man when he put all creation under the bondage of decay. And this would be a way to show all of us, in a way that safeguards our free will, that as things work in nature, it is impossible for non-living chemicals to form proteins, the building blocks of life. To use the example of Darwin's supposed warm little pond somewhere, that pond would have to be packed with millions of each of the 20 left-handed amino acids needed for life. And science also tells us that that little pond would also be packed with a near equal amount of right-handed amino acids. So, if you're going to believe this racemic mixture came together to form proteins, you must disregard the known effects of entropy and diffusion and believed that somehow the millions of left-handed amino acids were sorted out and isolated from the right-handed ones and separated with some sort of a barrier. Then, as time went by, while waiting for the left-handed ones to join and fold into proteins, something else must continually reach inside the barrier and selectively remove the left-handed ones that are racemizing into right-handed ones, because right-handed amino acids are toxic to the formation of proteins. If you believe this happened, you will have to do so using blind faith, because there is no evidence it could happen. God created you in His image so that you could live in fellowship with Him forever. He made the only way for you to come to Him through the cross of Jesus Christ. The prayer that Jesus made in John chapter 17 shows how much He desires fellowship with you. But fellowship must be based on free will, your free will that God will not violate. So, while remaining out of sight, He has provided ample evidence in His creation and in His Word for you to know that He is there and that He created you. Some of that evidence is in the form of biochemical discoveries like racemization of amino acids that scream that biochemical evolution is impossible. Our hope is that you will heed the evidence from science that will point you to your Creator and follow Lord Kelvin's advice when he said, "Do not be afraid of being free thinkers. If you think strongly enough, you will be forced by science to the belief in God."
Let's think about this from the argument that we're making here. Evolutionists and even CPT creationists, catastrophic plate tectonic creationists that reject the hydroplate theory, they want to say that this right side pile of blue and red is proof that it didn't come from life, that it was spontaneously made left and right-handed, because only life from DNA instructions through RNA will make it so it's all left-handed proteins, amino acids used to make a protein. And the reason why the evolutionists do it is obvious. They want to just say that these are the building blocks. Somehow these building blocks on the right pile, they come together on an asteroid, even though we can't even make them come together on Earth without human intelligence being devoted to certain mixtures of soup and electrical voltages and everything else. They can't make it. But somehow, in the vacuum of space, on a floating rock pile, they came this way. Non-HPT creationists, they want it to be a result of chemical processes instead of decay of protein life, because they can't explain how life got up into space. And they don't want to concede that, well, I guess amino acids can become alive. But all of that is blown away, as we emphasized in we're going to emphasize even today and last week, our last podcast. All that is blown away by finding bacteria and by finding nucleobases. You can't make nucleobases. So the nucleobases and the bacteria prove or strongly suggest that these mixed piles of amino acids are a result of the time decay of the left-handed into a equal 50-50 or close to equal 50-50. And besides that, so the evolutionists are teaching in all the classrooms now, all the Nova shows, everything else, they're saying, Bennu and Ryugu have proven that, this idea of panspermia, that the Earth was seeded with the building blocks of life, nucleobases and amino acids, and somehow that gave birth to life. I want the listener to think about this. They can't just have amino acids and nucleobases. You have to have the genius we've already talked about of these cellular machines that are sending the DNA out to go make an RNA. You need DNA to make RNA. You need RNA to make DNA. So how did that happen? You send the instructions out to go do all that miraculous, engineered, turning amino acids into a protein, and then you have to do all the proteins to come together to be something like a living creature. or even the proteins that are in a bacteria, or the living aspects of a bacteria. So they're counting on our ignorance. They're counting on our foolishness to believe their Santa Claus story, because there is so much to make life. Time and chance can't do it.
Now that we have a good way to answer the issue of finding both left and right-handed amino acids on asteroids Ryugu and Bennu, let's talk about how these living systems, trees, people, mammoths, how they were rapidly disassembled into their component amino acids, those amino acids then landing on Bennu or Ryugu and degraded over time into a combination of left and right-handed amino acids. So breaking down, disassembling living systems like that takes a huge amount of energy, and the water erupting out of the fountains of the great deep, as evidenced by the orbital paths of comets and asteroids, indicates a pretty good idea of the velocity that the water, water vapor, pulverized rock, and anything that was in its way is going to get hit with the force of this material coming up out of the earth. So just to give you an example of the effect of such rapid acceleration, I spent some time with AI and a couple of different conversion sites to make sure these numbers are correct, because they're pretty astounding. As you may recall, back in my bio, I told you how I was an F-16 pilot in the active duty Air Force and the Air National Guard. And the F-16 is an aircraft that's capable of generating a turn that will produce 9 times the gravity acceleration, 9 Gs. Those of you that may have seen the Top Gun movies can understand G-forces. It's the amount of force that's put on, for example, the pilot's body. And since at the time I weighed about 150 pounds, and a 9G turn, my body in that 9G turn all of a sudden weighs 1,350 pounds. And one of the quickest weight loss programs on Earth, when I was weighing 1,350 pounds, I released the stick and I was back to my original weight of 150 pounds. So that's an F-16. The escape velocity that the water would need to leave the gravitational influence of the Earth is 7 miles per second. That translates to 25,200 miles per hour. And I just picked a number for the acceleration time. I picked 1/4 of a second. It could be less, it could be more, but I think that's reasonable. So the answer that AI and a couple of conversion sites gave me, that if I, as a 150-pound person, were to be rapidly accelerated to 25,200 miles per hour in 1/4 second, I would experience 4,590 Gs, which would make me 688,500 pounds. That's the weight of a fully loaded Boeing 747 SP model. Now, in the fountains of the great deep, it's not all going to come out at 7 miles per second. Some of it will be less, some of it more. Some of the asteroids and the comets, their orbits indicate that they had to have a launch velocity of 32 miles per second, which is huge. You convert that into miles per hour, that's 115,200 miles per hour. Do that, accelerate to that in a 1/4 of a second, that's 21,005 Gs, which now my 150-pound body would weigh 3,150,862 pounds. There's no way that my body would be able to hold together. It would be crushed into its component parts. So let's take those numbers and take a look at, I picked a mammoth as an example. And the reason I picked a mammoth is I could have picked any animal, but because Ken Ham made a statement about Walt Brown's hydroplate theory in producing the material that became the comets and the asteroid, made the statement, Walt Brown says that mammoths were launched into space. And
how ridiculous that is.
And how ridiculous that is. So in this graphic, which was put together by our editor, and big shout out to our editor, Caleb. His superhero power is making really good editing videos and illustrations like this. But in this illustration, we have a mammoth is in a temperate environment. And the evidence of the frozen mammoths indicate that these are not cold weather animals, as depicted in textbooks showing mammoths trudging along in the snow. They've got fur. They've got hair. They don't have fur. They don't have erector cells or oil glands. And take a look at that nose. I lived in Duluth, Minnesota for a number of years, and you step outside on a cold day, your nose gets cold. How would you like to have a 6-foot nose hanging out there unprotected in a blizzard-like conditions as they're depicted in textbooks? That's not where they lived. The evidence indicates, based on food remnants in their mouths and in their stomachs, they lived in very temperate environments. The fountains of the Great Deep cracking open and bursting forth, and this unfortunate mammoth is about to receive a very rapid acceleration up to 32 miles per second. And so because of the tremendous amount of acceleration that flow would near instantly subject that mammoth to, is not just the acceleration, but it would be anything that's coming up with that impacts the mammoth or people, to include the dust, rocks, and water vapor. The cutting power of high-pressure water can be illustrated in machines that can use a high-pressure water stream to cut through very thick steel. So living things are subjected to this huge blast, force, acceleration, things hitting them, and it was rapidly disassembled into its component amino acids. And so whole mammoths don't get launched into space, but their component molecular parts, evidence indicates that those parts did. Not whole mammoths, their component parts did, disassembled by the forces of the fountains of the Great Deep. Dr. Feldman says there's no traces of plants or bacteria, only chemicals which may or may not have come from life. And as taught by on part 2 intro of the asteroid chapter number 6, they did find bacteria on asteroid Ryugu.
And the bacillus bacteria that they did find was not the result of a contaminated clean room. HPT and everybody else should really realize this is a fact. Why? Because how can they be sure they found amino acids and nucleobases if they are sure the clean room was contaminated with bacillus bacteria? In other words, they should have announced that the billion-dollar project, American billion-dollar project to Bennu and whatever the Japanese spent on Ryugu, sorry, we took all your tax money, but we couldn't make a clean room, and so we can't even tell you what we know we found. We think we found amino acids, but we also found bacteria. The bacteria was from a contaminated clean room, but the amino acids and the nucleobases weren't contaminated? They can't do that. So HPT, if the discovery of bacillus was a result of a contaminated clean room, then all space missions to collect samples from asteroids and comets should be canceled. We should be marching on Washington and say, hey, we want more food stamps instead of wasting billions of dollars in space. If you can't ensure a clean room with $10 million, then why bring samples from a $1 billion mission into an impossible to clean, not clean room? The bacillus bacteria survived about 5,000 years. This is hydroplate theory. The bacillus bacteria found on Ryugu survived about 5,000 years in a spore state, then recultured and reproduced in the lab when the environment was favorable, which is what bacillus bacteria do. When I was at Creation Research Society, one of the chemists that was giving a speech was talking about how robust bacillus bacteria is when it goes into a spore phase. It's another miracle. It's another miracle that the evolutionist time and chance God cannot explain. And I found this as an animation of what they were teaching at the conference. bacterial spore formation animation video. I couldn't find anything on this that told me how to do a copyright. So we're going to look at this as fair use, and we're giving you here the YouTube location for this that was produced by Study Force. But this is really another phenomenal engineered DNA, RNA regulated by the code of DNA and RNA miraculously telling this thing how to go into a spore phase and survive. And by the way, it's this spore phase that makes anthrax so incredibly dangerous that can come back after decades of laying in the ground.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
So, to repeat ourselves, how could anyone believe the extremely complex engineered biological system that allows Bacillus bacteria to survive the harshest of conditions could occur by time and chance? How could that happen? Answer, only the ones who are religiously devoted to their time and chance God. The same as we concluded with our last podcast. This is Darwin. If it could be demonstrated that any complex organ existed, Bacillus bacteria, which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. It is broke down. It is broke. But I can find no such case. I hope all the evolutionists that are watching this, I hope you can realize that you just found a case that you should break down the evolutionary lie and embrace the truth. And as we concluded with our last podcast, there are no, what Dr. Feldman said is not true. He says, because he's got to say it, because he can't, that was one movie line, he can't handle the truth.
You can't handle the truth!
He says there are no traces of plants, plant pieces is what Walt meant, or bacteria, only chemicals which may or may not have come from life. Not true. The scientific information from space, past and present, does not support Dr. Feldman's assertion again. So we continue to, we're not trying to convince people hydroplate theory is right. You don't go to heaven because you think comets and asteroids were born in the flood of Noah about 5,000 years ago. You don't even become a believer in Jesus and saved as a Christian to believe in a global flood at all, or that you were created. It really takes acknowledging that you want to have a relationship, to change your mind and say, I don't want to reject this God who created me. I want to change my mind and, one, confess that he exists, confess that I'm not God, confess that I've been deceived and I've done things sinful against him, and then say to God, God save me. That's what King David, when he had sinned, God, a broken and contrite heart, that's what you want. For us to come and say, Lord, Jesus, we want you to be the Lord of our life. We want to turn our life over to you. And we pray that that's what would happen. Bill, did you want to add anything on that? You know, the emphasis on the belief, what it takes.
Like Kevin said, it's change your mind. And you're going to have to change your mind about a lot of things, but ultimately change your mind about whatever it is you think you can do to save yourself from being separated from God forever. And change your mind and trust in the only one who can, because he paid for it in the past, and he said it's finished. And if you change your mind and trust him instead of yourself, you have eternal life, and you can know that you'll be with him forever. You don't have to fear death.
Amen. So who are you going to serve out there? You only have the time until you take your last breath to figure it out. You're either going to serve the one who loved you so much he sent Jesus and make a way for you to be saved, or you're going to serve the God of time and chance. Worship the creation instead of the creator. Read Romans chapter 1. It doesn't turn out well for those who go that route. God bless you, and it was a blessing for you to be able to join us on this 9th podcast, and we thank you for joining us. God bless you.
Thank you for viewing this episode of Hydroplate Theory University. We hope you'll return for future episodes. Please like, subscribe, spread the word, and be sure to visit creationscience.com, where you can find more great hydroplate theory resources.